Welcome to our first vodcast for the week of March 2nd. If you're joining me from the regularly scheduled classroom, then this is Monday, and we are thinking a little bit about the poster hypotheses. I probably am working my way through grading those right now and giving you feedback. Please turn them around as soon as you can if you do get feedback from me. But if your group just hasn't had a chance yet to submit a first draft of the hypothesis, please do so as soon as you can. Also, let's get those fever discussions running and rolling and rocking. We would like to try to finish them up by the end of the week this Sunday. I also will be holding my office hour today, and remember that that will be a special elongated office hour from 7:30 to 9 p.m. We will also decide whether we feel that an added office hour is necessary. We'll see how we do on the homework and getting through all of the concepts. In terms of labs, don't forget to get your dilution problems done, and remember that your TAs are more than amenable to helping with those. Hopefully by now you've already started to rock them through and you're feeling good about them, and I hope you've also started to really get moving on those skill assessments. The skill assessments are going to be due on the week of March 9th to the 13th, so please think about getting those done, getting those submitted, and being ready to show your TAs how much you've learned and how much mastery of each of the skills you have. We're going to dive right in, and we're going to remember that we left our coverage off last time by looking at the players in metabolism. And we had talked about reducing power and the high energy equivalency of reducing power in the form of NADH and FADH2 and even NADPH, which of course takes its electrons to power biosynthetic reactions, whereas the others that formerly mentioned to take their electrons to the ETC to power the synthesis of lots of ATP. Well, ATP, there you have it, the other major player in metabolism. Whether ATP is synthesized via another high energy molecule or whether it's synthesized in oxphos, we can recognize it as being an incredibly important energy currency. And in fact, the thing that I love about ATP is, as it turns out, and this always makes me feel better about life, the universe, and everything, ATP is wonderful because it's actually just really mediocre. So let's see what makes it so wonderfully mediocre. Looking at the molecule, we notice that it is adenosine triphosphate. We recognize the ribose sugar in the five-membered ring structure in all of its glory, with the hydroxyl, of course, at carbon 2 because it's not a deoxyribose. So we notice too that, of course, the adenine is attached at carbon number 1. So that is what gives it the name adenosine triphosphate. So the triphosphate, now this is actually where we want to spend our time. It's actually those linkages between the alpha, beta, and gamma phosphorus atoms that actually lead to the high energy of this molecule. And there are 3 of these linkages. 2 of them are the same. The other one is a little bit different. Let's begin with the different one. This one right here, you might recognize it as looking a lot like an ester bond, except for that it's a phosphorus atom here. So it's a phosphoester bond. This is pretty darn high in energy. In fact, it weighs in at 14 kilojoules per mole. Meaning that if you were to break this bond, right, the delta G naught prime of hydrolysis is negative 14. If we were to break that bond, standard state free energy release would be 14 kilojoules per mole. A lot of energy would be released. But guess what? Not as much energy as when the phosphoanhydride bonds are broken. So notice that these 2 bonds here are equivalent, and they are both worth more than we can even reckon at 30 kilojoules per mole. So notice that more than double the energy comes from the breakage of a phosphoanhydride bond as compared to the breakage of a phosphoester bond. So just breaking down those 2 linkages, wow, could that yield a lot of energy. That's why when ATP is converted to ADP and we break this linkage at the gamma phosphorus, that yields a lot of energy, approximately 30 kilojoules per mole. So one might ask the question of why are the phosphoanhydride bonds so high in energy? And to answer that question, we need look no further than, wow, how does this negative charge feel about this negative charge feel about this negative charge? They're like, whoa, howdy, you repel me, right? You definitely really repel me. You make me uncomfortable. And in that uncomfortable state, I am in a constant high energy state. So due to the steric repulsions in that molecule, we see a very high energy, very unstable molecule. And that is where that energy comes from. Quite simply, if these phosphates were to break and become individual inorganic phosphates alone, they would be more highly salivated with water and they would be happier, right? Less repulsion. They would be attracted to the water via ion dipole interactions, and everybody would be at a lower state, a little bit more chill. So that's how we could answer why are the phosphoanhydride bonds so high in energy. And if you want to just jot down, you should probably jot down more than just this, but you probably would want to at least include in this steric repulsion. Right? Charge repulsion due to the nearness of those negative charges. The other question that we need to ask is, noting that the triphosphate is what gives this molecule such high energy, we should ask the question of what about the other triphosphates, UTP, GTP, CTP? What about the energy that could come from those molecules? Well, it's the same. So in fact, ATP is not the universal energy currency because it's somehow better than those. It's just because it was evolutionarily selected for. So UTP, CTP, GTP, TTP, all of these are also very high in energy. So essentially the same, delta G naught prime, for these values, for these analogous phosphoanhydride and phosphoester bonds. So thinking back to ATP as the energy currency molecule, we know that when ATP is broken down to form ADP, we know that the high energy phosphoanhydride bond at the gamma phosphorus breaks. That yields approximately 30 kilojoules per mole. So we could write down that when these bonds are hydrolyzed, power is released, and we can see here it takes this simple equation of ATP plus water yields ADP plus inorganic phosphate plus energy, right? This can give off 30 kilojoules per mole that can be used to power energy requiring reactions, aka, said another way, when ATP's bonds are hydrolyzed, that is exergonic, right? It gives off energy. This could be used to power an endergonic or energy consuming reaction. So to better understand that idea, we have a table called the table of phosphoryl group transfer potentials, and I've adapted this table from another textbook and included some of the most interesting molecules on it. These molecules are things like phosphoenolpyruvate. We briefly met that molecule before and talked about the fact that it is wicked high in energy. But check this. Look, it weighs in at negative 62 kilojoules per mole. So in fact, that is so packed with power that there's literally enough energy in the bonds of PEP or phosphoenolpyruvate that in theory it could power the synthesis of 2 ATPs, so we often will abbreviate this PEP. But, of course, those of you who understand quantization of energy will understand that, in fact, this can only power the synthesis of 1 ATP because it really only has 1 phosphoryl group to transfer. So let's check out how I knew that this molecule had enough energy to make ATP. Recall that ATP has about 30 kilojoules per mole in those phosphoanhydride bonds. So if we wanted to take an ADP and an inorganic phosphate to make an ATP, it would require that we put in 30 kilojoules per mole. So PEP has 62. If it breaks down, it gives off 62. There's plenty of energy to power the needs of 30 to make ATP. Hey, look, there's also plenty in 1,3-BPG or 1,3-bisphosphoglycerate, negative 49. There's plenty energy to power the synthesis of ATP from that. Also, phosphocreatine. I was thinking, Ashley, you might have heard of phosphocreatine. This is a common supplement in the world of weight training. Phosphocreatine is often used to push. If it's consumed in the diet, the idea is to push the synthesis of phosphocreatine. So people will take in creatine and then try to make more phosphocreatine to serve as a reserve to make more ATP when they need the energy. So notice that phosphocreatine is a high energy phosphoanhydride that can make ATP by transferring a phosphoryl group to it. Even succinyl-CoA could provide enough energy to make ATP. So these molecules with these high phosphoryl group transfer potentials could transfer a phosphorus group onto ADP to make ATP. They could provide this molecule and enough high energy to make ATP. So let's write down the take-home message there is that these molecules can donate phosphoryl groups to ADP to make ATP. So in the process that requires energy to make and synthesize ATP, if a molecule provides that energy, so for example, if phosphoenolpyruvate is the source of that energy, right? If it provides that wicked high energy that is needed to make ATP, then we recognize the process by which ATP is made as being something called substrate level phosphorylation, because the molecule is a substrate, PEP, of an enzyme that transfers a phosphoryl group onto ADP to make ATP. So whenever a molecule is the source of the energy needed to make ATP, we call the process substrate level phosphorylation. Super important. Notice that I put that in red and I've also underlined it and I've also made it bold. Substrate level phosphorylation is the type of phosphorylation that takes a high energy molecule, uses the energy from something like 1,3-BPG or phosphocreatine or succinyl-CoA to make an ATP molecule. Now that totally differs from oxphos. Oxphos uses the PMF to make ATP. So substrate level phosphorylation is completely different. It uses a high energy molecule rather than the PMF to power the synthesis of ATP. Now we need to practice whether we fully understand how to use this table of phosphoryl group transfer potentials. Let's see if we can understand how we might figure out what the overall free energy change is for a reaction that is powered by something like 1,3-bisphosphoglycerate in which ATP is made. And that is what our next slide asks us to do. So use the table of phosphoryl group transfer potentials to answer the following question. In this reaction, 1,3-bisphosphoglycerate plus ADP goes to 3-phosphoglycerate plus ATP. Or said another way, high energy 1,3-bisphosphoglycerate phosphorylates ADP to make ATP. Hey, substrate level phosphorylation. So we definitely know that's taken place here. We can answer one of these really easily, right? B has got to be true. ATP is generated in this reaction via substrate level phosphorylation because a high energy molecule, 1,3-BPG, is giving a phosphoryl group to ADP to make ATP. So now we want to know what is the overall delta G for this entire reaction based upon the delta G's that we already know from the tabulated values. So let's write down the two pertinent reactions. 1,3-BPG is going to 3-PG, just abbreviating, plus an inorganic phosphate. So that's the inorganic phosphate that's getting transferred onto ADP. So here's our other pertinent reaction. ADP plus PI is going to yield, yay, you got it, ATP. Oh yeah. So let's write the delta G's for each one of these. Remember that we just talked about the fact that the delta G naught prime for 1,3-BPG getting broken down is negative 49 kilojoule per mole. Now for ATP getting made, why, that's going to take 30. So we need to make sure that we switch the sign on that and say that 30 is going to have to be input in order to make the ATP. Now if that ATP is getting broken down, then 30 would be released, but here it's getting made, so we're going to have to say we're going to have to input that. So let's add those up. Oh, and you get to cancel stuff here too, right? Because the inorganic phosphate was essentially transferred from 1,3-BPG onto ATP. So now we can do a little math, and Jonathan is saying to himself, this is minus 19 kilojoule per mole, and maybe he's already come up here and circled the overall delta G naught prime for this reaction is negative 19 kilojoules per mole. So it looks like our actual answer here is A and B only, right? That's our answer there. Okay. Sweet. So let's just briefly go back to this phosphoryl group transfer potential table and make sure that we summarize the usage of this table. And here it is in its full glory. Let's just go through this note. Note that the tabulated values are for hydrolysis of each metabolite. If the reaction indicates a synthesis, then we need to flip the sign. So for example, when we look at this reaction, ATP plus glucose equals glucose 6-phosphate. Okay, let's check this out. ATP has 30 kilojoules per mole in its bonds. Is that enough to make glucose 6-phosphate, which takes 14 in order to synthesize it? Well, when it breaks down, it gives off 14, but if it's going to be made, it's going to take 14 to make it. So if we think about that, we would recognize that ATP, if ATP broke down, it could easily power the synthesis of glucose 6-phosphate. In fact, the overall delta G naught prime for that would be negative 16 kilojoules per mole, very exergonic. So yeah, there's plenty of energy in the bonds of ATP to make glucose 6-phosphate. So practice with this table. Try thinking your way through, could this molecule make this molecule? Could this molecule make that molecule? And what would be the overall delta G naught prime for each of those reactions that I'm envisioning? Marvelous. We're going to now move on, and I'm so excited because we get to start our coverage of metabolism in its full glory. I am so jazzed to dive into the depths of metabolism, looking at all of the nuances of each one of the pathways. But before we can cover those nuances, I want to begin with a big picture view and an analogy. I find that talking about metabolism is a lot like taking a trip through a canyon. Every year in the spring, we take the ski team into the canyons, and the way in which you have to approach a trip like that is that you enter a canyon from the top portion where it is much less deep. And as you make your way down into the canyon, you have to progressively get to a lower and lower and lower level until you're at a level you see in this picture where the canyon walls are 500, 1,000 feet sometimes above you. So the journey requires that at each level, you may have to make several rappels, and you may have to have a way to get your rope down to follow you down in order to get down these big ledges or pour-offs, and you can see a pour-off here in this picture. I find that this is very much like metabolism because in metabolism there are certain steps that you take that if you wanted to, you could turn around and those steps could be reversible. Just like in the canyon, there are some places where you could simply turn back around and walk back up. But if you've just come off a huge pour-off, you're not very well going to be jumping back up that pour-off. The same way in metabolism, there are certain steps that we call metabolically irreversible. That is, they cannot be simply reversed. And in order to get backwards in steps like that, a completely different bypass reaction would have to be run. Just in the same way, in the canyon, if you wanted to get back up those pour-offs, you would sure as heck have to find a completely different bypass route in which you could actually climb up and around. So with that in mind, I want you to continue to think of the canyon journey analogy, and I want to maybe approach this from the same standpoint that you would approach a journey into the canyon. That is, you would begin first, absolutely, and you would have it with you. You would begin by looking at a map, and you would always have a map. And so that's how I want to begin our journey into the metabolic pathways, is by looking at a map. And I kind of consider this first slide, even though it looks overwhelming and it's for full form, same way with a map, right? If you looked at a map and you took the whole thing into account rather than just looking at the place that you wanted to go, it would be kind of overwhelming. So we're going to begin with this big map picture view, and then we're going to zoom in to every little pathway and deconstruct all of the nuances. Let's start out with what we know to be a beginning energy source for metabolism, donuts, right? That is to say, starch. Starch, and of course we know also storage glycogen that may be in a microbial cell. It might be found in a glycogen storage granule. We know in us it's found in our liver. And these polysaccharides, these carbohydrates, can get broken down into their glucose subunits. Remember that glucose is a 6-carbon sugar. We've drawn it a number of times now, so everybody is way familiar with it. And the first thing that's going to happen to glucose is that it's going to get broken down in what I like to say in this image is the black box of glycolysis. And you can see here just showing glucose going into glycolysis. And we know that in glycolysis, glucose is going to get ripped and stripped apart. That is, it's going to get broken down from its 6-carbon form into 2 3-carbon molecules called pyruvate. However, we also know that as glucose is broken down and ripped and stripped of its high energy electrons, we know that it yields other forms of energy. For example, ATP, right? So we know that one of the things that's going to be coming out of this black box of glycolysis, or rather it's pink, right, is ATP. We also know that the high energy electrons that are getting stripped off of glucose aren't going to run around naked in the cell. We've talked a lot about that. Instead, they're going to have these wonderful carriers. NADH, being the reduced form of nicotinamide adenine dinucleotide, is going to be the carrier for electrons coming out of glycolysis. Now, we could think about the formation of the 3-carbon pyruvate molecule as being sort of a point in, by analogy, in our canyoneering journey, where we're hit a place where there's actually 3 separate different canyons going in different directions. We could go down the one to the far right, down the one to the far left, down the one to the middle. There are a lot of choices. We sometimes say that pyruvate is the molecule of many fates. We talk specifically about 3 or 4 of those possible fates, though, and we will talk and look at those with time. The one that I might mention now is that, in fact, the 3-carbon pyruvate could actually get usurped at this point and be used as a Lego, a building block for anabolic reactions to build up other molecules, such as the amino acid alanine. That's true of other molecules in glycolysis. Note that some precursor metabolites may be derived from the glycolytic pathway. One of the possible fates of pyruvate is to keep on getting oxidized, that is, ripped and stripped of even more high energy electrons. Now, pyruvate is already a pretty oxidized molecule, so when it gets oxidized again in a step that we often call the transition step, this step makes more high energy electron carrying NADH, but it also creates a 2-carbon product, 2-carbon acetyl. Now, if we know that pyruvate is a 3-carbon molecule, you guessed it. This is the place where we finally get that first fully worn out, totally used up carbon dioxide. So it's really at this point that the first CO2 is liberated from the catabolic pathways. So CO2 getting released, 2-carbon acetyl group going on to be further ripped and stripped of its high energy electrons. This is where the favorite pathway, maybe many of you have talked about this pathway before, maybe you know it as the Krebs cycle or the TCA cycle or the citric acid cycle. It is the cycle of many names. But I would argue that in fact, if we were to give this cycle a true description of what it does, we would call it the serious reducing power generator, because this pathway oxidizes the 2-carbon acetyl group absolutely and completely. And so in the end, we know that the TCA cycle is going to be spitting out a ton of CO2. In fact, 2 CO2s for every acetyl group, because that acetyl group, remember, is 2 carbons in size. So there would be 2 fully worn out, totally used up carbon dioxide molecules that would get generated from that TCA cycle. We also know that the TCA cycle is going to be further oxidizing the 2-carbon molecule, therefore giving off tons of reducing power in the form of NADH, but also in the form of the other one, FADH2, which can quickly give its electrons to Q to make QH2. So tons of reducing power coming out of the TCA cycle, CO2 coming out of the TCA cycle, and you guessed it, our good friend ATP also deriving out of this black box of the TCA cycle. So as we look at this portion of our map, we see more reducing power getting generated, we see ATP getting generated, and more potential precursor metabolites, more Legos to build up in anabolic pathways. So we'll look, I promise, at the depths of the TCA, but at this point, we need to trace the journey of all of that dang reducing power. So at this point, notice this, we have gotten reducing power all over the place. An NADH that comes out of glycolysis, we'll look at stoichiometry later to talk about the number of NADH. We get NADH coming out of the transition step. We get NADH coming out of TCA. Notice we also get FADH2 reducing power coming out of TCA. And remember that this reducing power runs its electrons to the ETC, where it can get passed from one member to the next member to the next, protons get pumped, PMF gets formed, and ATP, right? That whole jazz, as I can already see Levi saying to the computer screen, he's like, hey, that's aerobic respiration, right? And it might be aerobic, it certainly is aerobic in our bodies. So let's look at respiration, where all of the reducing power delivers its electrons to the electron transport chain, and we know that that can be utilized to make ATP. Now, you might even be, if you're thinking with a real sexy mind right now, you might be thinking, I wonder if the ATP that comes straight out of glycolysis and straight out of the TCA, I wonder if that's different than the ATP that comes out of respiration. So keep entertaining that question because it's a very good one. But I have a feeling that some of you are thinking, perhaps some of you are just completely distracted looking at this box over here, fermentation. You're like, yes, I wish I was studying fermentation right now. If that is the case, do note that one of the possible fates of pyruvate, remember we said it's a molecule of many fates, one of its potential fates is in fermentation. Now, this is true when respiration is not going. So let's just remind ourselves that if respiration is not happening, if there is no ETC, if there is no terminal electron acceptor, for example, in us, if there was no oxygen, or, hey, since we're talking fermentation, let's talk about yeast. If yeast had no oxygen to serve as a terminal electron acceptor, that would mean that respiration would not occur. That would mean that NADH and FADH2 would have no place to take their electrons. That would mean that TCA, middle name reducing power, would not occur. It also would mean that the transition step would not occur, which would leave our cycle very little one here at glycolysis going into fermentation, and we would see that the fate of pyruvate would be to serve to accept electrons from NADH because it's worthless, it has nowhere to go, it can't take its electrons to the ETC. So all that would happen is pyruvate would serve, or a derivative of pyruvate would serve as an acceptor for those electrons, allowing for the regeneration of NAD+ and allowing glycolysis to keep on going. So in a yeast cell that cannot utilize respiration, all it can do is fermentation. And the only ATP that that yeast cell can make is in glycolysis. So when we make wine in lab, I like to say that when we put the stopper on the bottle and we starve the yeast of their oxygen terminal electron acceptor, those little yeast cells turn around and give us the finger. Why? Because all of a sudden they went from being able to make massive amounts of ATP in respiration to being able to make only a little bit of ATP in glycolysis. So perhaps Anthony is completely distracted from all of this and he's thinking to himself, Rachel, aren't there other fuel sources besides glycogen and starch? I mean, personally, like with all my weight training, I tend to live on almost an entirely protein diet, right? I don't eat very much starch in my diet. So what about proteins? Can they serve as an energy source? And maybe Zeri is like, Rachel, you always keep talking about donuts being a source of starch, but really, aren't they packed full of triacylglycerols or fat? True. So let's talk about these other fuels. Triacylglycerols are absolutely the most packed with power. So when a fat gets broken down, gets oxidized, ripped and stripped of its high energy electrons, it too generates lots of reducing power. The 2 carbon units from the triacylglycerols from their fatty acyl side chains enter into catabolism as acetyl-CoA. They then run the TCA cycle, generating even more reducing power. So fats are packed with power in the form of reducing power, and they enter in and are primarily used in the TCA cycle. You can think about what that means, about what a cell has to have in order to use fats as a fuel. And Anthony, you're right. Proteins can get used as an energy source, though it's important to note that proteins are the only toxic fuel that we take in our diet. Why are they toxic? Well, because they give off ammonium. This is ammonium here, has to be cleared in the form of urea. So we have to deaminate these amino acids in order to take them in. So amino acids can enter into catabolism, and there are a variety of places in which they do so as 3-carbon pyruvates, as 2-carbon acetyls, and as intermediates of the TCA cycle. But they are, as we said, the most toxic of all fuels that we might take in. This, I realized, was a very big picture map. And now our goal is to jump in to the heart of the pink box of glycolysis. So get ramped for the glycolytic pathway.